Thank you, sir. Yes. Okay. Okay. So we'll be starting sharp at 7.
So fine.
All the participants who have their microphones and video on, please turn them off. Hello, everyone, and welcome to our session today at Progress and Prospects in Biology. Am I audible to everyone? So, can you hear me?
Yeah, I can hear
Yes,
you.
you are.
Okay. Welcome, everyone, to Progress and Prospects in Biology. Welcome to today's session. We are delighted to have one of the leading RNA biologists of our country today with us, Dr. Suvendra Nath Bhattacharyya. To all those who are joining us today, welcome. And I'll just mention a few housekeeping requirements. Please mute your audio and turn off your video. Please do not share your screen at any point of time during the talk. All this is required to bring you uninterrupted streaming of the webinar. Please feel free to drop your questions in the chat boxes. Our team will be monitoring them and we will address them at the end of the talk. For the feedback link to get the e-certificate, that will be shared during the session. And now I will turn it over to our mentor at PPB, Professor N.R.I. Banerjee, for the welcome address. Ma'am, over to you, ma'am.
Thank you, Rupkatha. And a very warm welcome to all the participants in today's webinar. Welcome, Shubhendra. We are delighted to have you on our webinar.
Thank you.
When Rupkatha introduces our today's speaker with the very brief bio sketch that he has provided us, you will know that he is quite extraordinary in the sense he has achieved at a young age what most scientists aspire to achieve for decades. Probably the most important attribute.
In 2015, he received the Nasi Scopus Young Scientist Award and the Shauna Jayanti Fellowship. IIT Roorkee awarded him the Khosla National Award in Science in 2018. He also received the AAAS Young Scientist Award, the INSA Young Scientist Award, HFSPO and EMBO Long-Term Fellowship. He was also awarded the CDA Award of HFSPO and the prestigious International Senior Research Fellowship of the Wellcome Trust, London. Dr. Bhattacharya is a fellow of the National Academy of Sciences, India, based in Prayagraj, and the Indian Academy of Sciences, Bengaluru. With that, we will begin. Once again, a gentle reminder, please keep your audios muted, please keep your videos turned off, and feel free to write your questions in that box. We will be addressing them later. With that, over to you, sir. Thank you, sir.
Thank you, Rupkatha and Ninadi. It is a great pleasure to be part of this forum and share some of the work that we have already done in this microRNA world. And as Enadi mentioned, I will try to highlight a little bit of my journey that I have started after my MSc in biochemistry that I have done with the University of Calcutta in the biochemistry department in Maligan Science College. And I today I'm going to talk a little bit on only one aspect of my research, that's the microRNA movement. So what I mean by the movement is nothing to do with any, I'd say, uh, any kind of movement that we are used to know about in society, but it's all about a journey. So that is the mighty regulation of a tiny regulator. So I'm going to introduce why it's a mighty regulator and why it's a tiny regulator as well. Uh, so as Rupkata mentioned, that I am from the CSIR, Indian Institute of Chemical Biology, Kolkata, and I have a lab in the RNA biology aspects that we do. So in the career path, I'm an RNA biologist forever, starting from my PhD journey, which again started in 1998 when I joined Dr. Samit Ardho in the Institute of Chemical Biology with the exciting group there who are doing some kind of interesting research on one particular parasite called Leishmania donovani. In that particular type of work, that was a very fascinating kind of work that we are working on, the mechanism of mitochondrial tRNA input. So, as we know, the mitochondria has their own sets of tRNA and some mRNAs also that are getting translated. Almost 13 warps are there. But there are certain species like Leishmania, this kind of species that are there in the protozoan world. They don't have their mitochondrial tRNA there. But how they sustain their mitochondrial translation? So what they do, they import the tRNA that are encoded by the nuclear genes, and this tRNA input is essential for their survival. So there was no much information was there before I started the work there. So we worked on the mechanism how the mitochondrial tRNA input occurs in the leishmanial parasite. So later, I moved to the for my postdoctoral research in the Friedrich Mission Institute in Basel, Switzerland, with Professor Witold Filippo, which is a pioneer person in the microRNA world. Where I worked on how these small microRNA that I'm going to introduce a little bit later actually work as a repressor of translations or gene repressor to mediate the gene regulation process. So the mechanistic angle of that thing was get revealed at that particular time during this 2004-10 time, and I was there in his lab and exploring the mechanism of this kind of microRNA-mediated gene repression process. So later, when I started my lab in 2008 as a principal scientist in CSIR IICV, a very premier research institute of this country, where we work on the mechanism of microRNA activity regulation in mammalian cells. So as I mentioned during my postdoctoral research, I had interest in the mechanism by which the microRNA acts. Now we like to explore how the microRNA are getting regulated, so that the mechanism of microRNA activity regulations in the mammalian cells and its importance in gene expression process. So I'm going to introduce how the gene expression getting controlled. We all know about the central dogma where the DNA get transcribed into the RNA and then protein get translated from the RNA. So this process, the protein to RNA process is not reversible, but there are certain players that can influence this kind of DNA RNA protein biosynthesis pathways, and these are microRNAs. So these are basically small RNA that can regulate on RNA or even on DNA to affect the translation process or generation of protein by regulating the transcription and translation process, primarily affecting on the translation process. And the protein that they generate by the RNA is actually act on the microRNA itself and regulate their expression pattern as well. So you can think about that these are the tiny way of regulating the gene expressions, but this is a very mighty way of regulating as well. So post-transcriptional gene regulation in eukaryotes is a little bit more complicated than what we can think about. So there are 5' UTRs region and 3' UTR region or untranslated regions, and there are coding sequence that encode the protein part of the gene. So when we think about the different protein factors or other kind of factors that interact with these UTRs, one can think about they are probably there to support the translation process, but they are more than that. They can interact in a dynamic manner with the mRNA and can affect the translation stability and the localization of the mRNA. They could be kind of regulatory protein, they could be scaffolding protein, there could be accessory proteins. They all affect the process of gene expression by targeting either translation, stability, or the localizations from the of the mRNA. So we can think about an mRNP code that's about the RNA and the protein that they can generate this kind of code. So in these connections, microRNA is an important step. MicroRNA are 22 nucleotide long regulatory RNA that are transcribed as a precursor form, the primicroRNA, inside the nucleus, and then processed by DGCRA DORSA to a pre-microRNA intermediate. And these intermediate then exported into the cytoplasm, where they matured by the enzyme complex DICER and TRBP to generate the mature form of the microRNA, and the stand that encode the microRNA get incorporated into the Argonaute proteins, and the Argonaute microRNA complex then binds the target messages with imperfect complementary or perfect complementarities. And when they binds the target genes, they can replace the translations and ensure the degradation of the target genes. So you can think about there are thousands of microRNA operates in humans, and as they are higher in number and present in almost all cell types of humans, there are 50% of the human genes, or more than 50% rather, are predicted to be the microRNA regulated. As majority of the genes are under microRNA regulations, they regulate the development, differentiation, apoptosis, almost all processes of our life that we can think about. And their expression is also dysregulated in the disease conditions, as they are important regulators. And these microRNA also have some specificity of expressions depending on the different developmental stage and or the tissue type we are thinking about. So there are specific microRNA that are expressed only in brain or liver or muscles. But there are also some microRNA that are ubiquitously expressed in all over the tissues, like RED7 is one of the microRNA of that kind. So this microRNA depression could be also reversible. So as we can think about this microRNA depression occurs, they are taken to some kind of granules or bodies called P bodies, these PBs. So these are kind of phase separated entities in the cytoplasm of mammalian cells, where these microRNA repressed messages can be stored along with the microRNA. And then under specific circumstances like the stress response, there are certain proteins that relocate from the nucleus to the cytoplasms and can act on the mRNA and bind the mRNA and de-repress the microRNA actions. This happened like in human hepatocyte under stress condition, the one gene CAT1, which is under the microRNA 1 to 2 repression, get relieved under the stress condition by the help of this protein HUR, and RNA get relocalized from the P body. So that improved the reversibility of the microRNA repression process. On the other hand, there could be other kind of factors. In this case, HUR is a negative regulator of microRNA action that will inhibit the microRNA actions. But there are can be factors that are the positive regulator of microRNA actions. That means when they bind the UTR, they can help the recruitment of the microRNA and the Argonaute complex, the microRNP complex, to the target sites. In that way, they influence positively the microRNA-mediated repression. That means there will mean that there will be strong repressions when this protein factor bind along with the microRNA on the 3' UTR. So how this factor that could act as a positive or a negative regulator gets recruited? Yes.
Sir, very sorry to interrupt, but could you please share it again, because someone had shared the screen and the slides are not visible.
Okay, I'll just...
Please share it again. Sorry for the interruptions.
No issue.
See the slides.
We like to highlight the fate of the microRNA. So one about think about these factors that are there to positive or negatively regulate the fate of mRNA can also influence their stability and activity on the mRNA itself. But there could be some other aspects that are getting affected by the other kind of cellular pathways that affect the microRNA stability and expression. So one of the factors that govern the microRNA level is the exosome, small export of microRNA. So exosomes is a special class of extracellular vesicles that are of 30 to 90 millimeter vesicles secreted by a wide range of cells that contain both proteins and RNAs, including the microRNAs, and they are derived from a very complicated process through an endocytosis process. Like the early endosomes, mature late endosomes, and then to the multivesicular bodies that contain several small vesicles within the vesicles. And when this multivesicular bodies fuses with the plasma membrane, they releases the content into the intercellular space of the microRNA present in that particular cells to increase the microRNA level there. So that's why there's a very interesting way of regulating gene expression not only in the donor cells but also in the recipient cells. So when he mentioned that, it has emerged recently about the importance of the microRNA mediated exosomal export as a regulator of gene expression in different contexts. So one context that I am going to highlight in this particular slide is the role of this exosomal export of microRNA in the cancer development and or tumor development. So we can think about the donor cell excrete the microspecific microRNAs as it named here. They're going to the tumor cells or some other kinds of cell to promote the tumor growth or angiogenesis or metastasis or drug resistance processes, as well as it's going to the other kinds of cell present in the tumor niche like the immune cells and affect their function as well. Also, the cancer-associated fibroblast also receive specific sets of microRNA and that also affect their function. So intercellular microRNA transfer between different types of cells can create the tumor microenvironment which is necessary for the cancer progression. So we talked about the importance of the exosomal export of microRNA so far, but the one important aspect we need to know if we like to use this knowledge about this exosomal export of microRNA in a betterment of disease management and health, we need to know the regulation of this kind of export process that's happening in the mammalian cells and the microRNA between two kinds of cell with a different level of microRNA. So we can say that insulin-like growth factor 1 prevents the miR122 production in the neighboring cells to curtail its intercellular transport to ensure the proliferation of human hepatocytes cells. So this is in which you are facilitate the export process of microRNA. I'm not going over. So that is the paper.
Sir, I'm sorry to interrupt, but the slides, I think somebody had presented the screen. Could you please share the slide again?
I'll try to do so.
Sorry for the interruption, sir. It's a request to all participants, please do not share your screens.
Problem, maybe.
Is there a problem with the presentation?
Just checking something. I can't see it.
Sir, it will be better to reshare the screen.
Yeah, I'm trying that. I'm not getting the option.
Yes.
So that's the only problem I find.
Okay.
Okay, sir.
Yeah. So again, it's visible, I hope.
Yes, sir. It's showing
Yes,
the
yes, sir.
presentation.
Yeah.
Yes, sir.
Thank you, sir.
Okay, so I'm just recapitulating
Sorry.
what we are talking about. Under the fate conditions, the repressed message is there. Under the start conditions, the HUR get relocalized, replace the microRNA from the microRNP from the UTRs, and as well as it replace the microRNA from the protein argonaut and get bound to the microRNA. For the export to occur via the exosome, the microRNA unbinding with the HUR need to happen. So it happens by a post-translation modification of the protein HUR, and that is ubiquitinations. That ubiquitinations cause an extracellular export of microRNA in the mammalian cells. So as we can imagine that the export of microRNA is happens, so they are going to the intercellular space or in the blood flow to affect different process. So they can act as vesicular hormones. They can affect the intraorgan transfer of signals via the exosomal microRNA. So in this process, it is also important to highlight that export of this miR-122 from the high-fat export liver cells is very important to activate the macrophage present in the liver tissue, and that causes the metaflammation process. So that's the work we are working right now, and we'd like to find the link that how these hepatocytes release the microRNA, what effect the microRNA release by the hepatic cells when they exposed to high fat, and how the macrophage, the Kupffer cells and other kind of macrophage cells present in the liver get activated through the process. So we have talked about the reversibility of microRNA-mediated depressions during during the starvation of the hepatic cells. Now we are talking about how the reversibility of this microRNA actions in another context called macrophage activation context. So macrophage, when exposed to lipopolysaccharides, bacterial-derived molecules, they get activated. They activated through a different kind of activations process through transcriptional upregulation of certain genes like TNF-alpha, IL-1 beta, IL-6. But this is not the all case. The transcriptions there are something happens even before. So to do that, you have to think about the expression of those mRNAs that are supposed to be repressed by the microRNA during the activation process. There are a number of microRNAs that can act on these different cytokines that need to get expressed during the activation process of macrophage. So how the cytokines are getting de-repressed from these microRNA-mediated actions is not known. So we are going to talk about an LPS-induced reversal of microRNA action in activated macrophage. So if you use the reporter with the microRNA binding site in the UTR, we find within the first few hours of LPS stimulations, there is a loss of microRNA activity, and this loss is very much global. It also actually happened for all the microRNA we have tested. So there is a reversal of microRNA-mediated repression occurs, not only with this reporter mRNA repression, but also with the mRNA, cytokine mRNA as well. And this repression occurs because of different things happening there. So it happens that there is no change in the argonaut protein during this activation phase, but there is a loss of microRNA binding, and the target RNA binding happens with the argonaut proteins. And these actually, because of the LPS treatment, restrict the target mRNA binding to the argonaut in the activated phase. And later we find there is a loss of microRNA binding as well, and that actually happened because of the phosphorylation. So we have a paper published in EMBO reports on that particular topic, where we have shown that LPS treatment de-repress the microRNA activity in the macrophage, loss of microRNA from argonaut occurs, phosphorylation of the argonaut occurs, and that actually causing the microRNA unbinding, and the defective argonaut phosphorylation resulted reduced inflammatory response. So the phosphorylations that happen through a tyrosine kinase, because it's phosphorylating a tyrosine residue in the argonaut protein, is important for the microRNA-mediated de-repression process to occur. Later, we also find the process get de-repressed in a different manner because the phosphorylated argonaut again become dephosphorylated in the longer exposure of LPS. That means there must be a phosphatase act on this phosphorylated argonaut 2 to reactivate them, to repress the extra cytokine produced because of the derepression of the microRNA. So phosphorylation of Argonaute 2 and the microRNA unbinding is an important process that is reversibly get regulated in the macrophage during the activation process. In this context, HUR also play an important role because HUR also have the capacity to bind the RNA with AU sequence, the mRNA I mean. So cytokine mRNA having the AU sequence are the targets for HUR, and HUR facilitated the translation of this pro-inflammatory cytokines like TNF alpha, IL-1 beta, and IL-6 because they have the AU sequence. So what happened to the microRNA? We find that LPS stimulation export the microRNA like LET7 via the exosomal export process, and that is associated with an increase of HUR level. But if we use a drug, this DW4 which blocks the export of microRNA, it affects the expression of cytokines, which is induced by the LPS, but cannot get induced in the presence of GW469. That means the exosomal export of this microRNA important for the cytokines induction. So does HUR have a role in the LPS-driven microRNA export process also? So we did the similar kind of experiment we did earlier, like the depletion of HUR and expression of HUR, and find that similarly that the TNF alpha, IL-6 or IL-beta expressions also getting affected by the SI-HUR treatment. Similarly, the cellular micron 155 levels also get increased in the HUR depleted conditions. And then we find that HUR is sufficient because if we express HUR, even in the non-LPS treated cells, we can see some increase in expression of cytokine like TNF alpha and other kind of cytokine, pro-inflammatory cytokines. So ectopic expression of HUR increases the pro-inflammatory cytokine expression. So what happens to the HUR-driven anti-inflammatory response observed during a pathogen invasion? So far we talked about the LPS-mediated activation process, but during the pathogen invasions, the macrophage inflammatory response get oppositely regulated. That means the anti-inflammatory response pathway should be there to take care of the pathogen infection process. So how the HUR-mediated export process get affected? So to test that, we use the one very favorite model of us, the Leishmania infection process of macrophage, because Leishmania is a pathogen that in the human system, when they enter through the saliva of the fly during the biting, they are taken to the liver, and the covert cells is in the liver is the primary site where they get infected, and they replicate. So Leishmania donovan is a causative agent of visceral leishmaniasis, and in the tissue macrophage, it upregulates the IL-10. The anti-inflammatory cytokines, and in that way affect the pro-inflammatory cytokine expression. But as HUR is there, it should try to express the pro-inflammatory cytokines. So what the parasite did, it reduced the HUR level. So during the infection process, you can see there is a drop of HUR level, and with the more infection in different strains of L. donovani, L. major, or L. mexicana in raw and primary cells, we have the similar results that there is a drop of HUR. On the other hand, this HUR decrease is getting prevented by the AMG132, a drug that inhibits the proteasomal degradation of HUR, and that not only restored the HUR level, but also restored the TNF alpha expression in the infected cell. And we have similar kind of HUR decrease in the mouse model as well of the infection in the 10 days and 35 days of infections. Leishmania downregulated the HUR protein. So there must be a protease that is induced by or expressed by Leishmania that should target the HUR for its degradations to ensure the suppression of the TNF alpha-like mRNA that are under the HUR regulation. So Leishmania express one important metalloprotease called GP63, which is a zinc metalloprotease, and that is very much known to be important for regulating different cellular host cell proteins by targeting them for degradation. So does GP63 also target HUR? So we use the purified HUR or in a lysate of cells and treat them with either the purified GP63 from the leishmanial membrane or crude extract of leishmania. In both cases, we can see a concentration dependent decrease of HUR level, and that can be rescued by orthophenantholi, a drug that chelates the zinc, because GP63 is a zinc metalloprotease. So if we chelate out the zinc, we can recover the HUR level. That means the protein get inhibited. And the similar observation we have in the in vivo when we use the GP63 encapsulated liposomes and inject them in the mice to see the decrease of HUR in the liver. So does HUR restoration of the HUR affect the infection level? So what we did, we expressed the HHUR in the infection context in the raw cells, and we see that the HHUR transfected cells, the number of leishmania internalized per cells get dropped, as well as we can see the restoration of the pro-inflammatory cytokine expression like TNF alpha and beta, decrease in IL-10 expression. And the microscopic data also suggests the same, the clearance of leishmania from the HHUR expressing cells. So we can see the LD counts also reduces upon the transfection of the HHUR expression plasmid. And we can also see that HHUR transfection leads to TNF alpha mRNA level increase and also IL-10 level decrease in the treated animals. So we can come to the model that the GP63 is a protein that targets the HUR, and by that way it reduces the pro-inflammatory expression, support the anti-inflammatory expression of the macrophage cytokines profile. But if there is an excess HUR that can overrule the GP63's presence, then the balance can be shifted from a anti-inflammatory to a pro-inflammatory nature, and that can clear the parasites. So that was a paper we published in the EMBO Molecular Medicines, where we talked about how the microRNA exporter protein HUR clears the internalized pathogen by promoting the pro-inflammatory response in infected macrophage. That was recently published in 2020. In this context, I just talked about only one part of the story. The story was not that simple as it seems. There are other kind of protein and RNA get expressed during the infection process, and one of them is a phosphatase called PP2A. Work from the other labs in IICB, Dr. Pijus Das lab actually worked on that to identify the importance of this PP2A, which is getting induced by the leishmania upon infections. And this PP2A is a phosphatase that act on phosphorylated argonaute 2. And I talked about this phosphorylated argonaute 2 when I talked about the LPS-mediated activation process of deactivation process of the microRNP and the inflammatory cytokine expression. That get reversed later, but this PP2A is actually induced by the leishmania to restrict this process of microRNP deactivation to ensure the repression of the cytokine by IL-1 beta and TNF alpha by the microRNP. On the other hand, as they secrete the GP63, this GP63 targets the HUR, and this HUR could then act on the phosphorylated argonaute 2 as well as on the PP2A itself. So there's a crosstalk between the PP2A and the HUR. And on the other hand, it also export out that getting uncoupled due to phosphorylation of argonaute 2 to reduce the microRNA load in the cells. So in both ways, the IL-1 beta and TNF alpha expression as well as IL-10 expression get regulated in a reciprocal manner by the protein HUR and the PP2A. While the HUR favors the pro-inflammatory cytokine like IL-1 beta and TNF alpha expressions, PP2A doing the opposite. So in that way, the balanced expression of these two proteins during the infection process or LPS stimulation process play a major role in controlling the nature of the inflammatory response shown by a particular cell. So that may not only hold true for the leishmania infection context, but it also very much possible in the other context of infections, including the viral infections and other. So what is the importance of this finding? So if we find that if you have a strain like leishmania resistant to the anti-leishmanial drug like antimonial drug that usually kills the leishmania, there are resistance form of the parasites that are resistant to that drugs. So there is no much way to cure them. So one of the way we can think about to play with and identify new pathway that may get affected. So in this context, when we use both the ocadoic acid as an inhibitor of PP2A that the leishmania induce and HHUR that are targeted by the parasite also, we can find there is an interesting way of curing the parasite. So if you have this pathogen which is resistance, we find just expression of HHUR doesn't have much effect on the clearance of the pathogen. Neither we have a just treatment of ocadoic acid could have an effect on the leishmanial count. But when used together, they are very much successful to reduce the load of leishmania that are resistant to the antimonial drugs. So they have a strong effect on pro-inflammatory response to induce when they are used together, and IL-1 beta level also get increased. So in this case, one can think about just shift the balance to a more pro-inflammatory nature by applying the ocadoic acid and HHUR. One can clear the more deadly pathogen like resistance form of the parasite leishmania. So they are very much in relation to each other. For example, the IL-6, one of the cytokines we have talked about, is very much in a mathematical relations with the miRNA expression. In this case, miRNA LED7. On the other hand, the relative level of IL-6 is also in a relation with the HUR concentration and the PP2A concentrations. So we did some mathematical modeling out of the data we had with the expression of HUR and PP2A in macrophage, and they are in this kind of relations. What does exactly mean? Don't ask me. I'm not an expert of that, but this is done in the collaboration of Krishnananda Chattopadhyay's lab of CSR ICB to find the relations between this kind of proteins and the microRNA and the cytokines. So it may be useful to explore in future in a systemic approach when you are talking about that. In the last few minutes, I just try to highlight another important aspects of the exosomal export process. So far, we talked about the importance of the export process to occur, how the process gets regulated by the protein HUR, how the cellular context like confluency and growth status of the cell can influence the process, and how the pathogen infection may alter this kind of export process to affect the gene expression in the donor cells. But what about the recipient cells? But the recipient cells should receive those exosomes that are released by the donor cells and should get internalized there. And therefore, the fate of the microRNA inside the recipient cell is a very much unknown territory. So for example, from in which the microRNA is transferred to the recipient cell, how the loading of the recipient cell RNA2 protein occurs with the received microRNA, the site of localization and interaction of the target mRNA in the recipient cell with this foreign RNA that are entered there, the route of internalization of the microRNA in recipient cells, and the role of other cellular organelles in the process that are all unexplored. And recently, we have done a work to explore these pathways. And I just summarizing some of the finding we had on that particular things. So we have identified that dynamin 2 is very much important. So for this, for the uptake of these extracellular microRNAs, we also find the importance of the RAF proteins like RAF5 and RAF7 in the internalization process of the microRNA. And later we find that when the endosome matured, they actually change the pH from a higher to a lower pH before the fusion of the endosomes occurs with the lysosomes. So the pH drops actually ensure the fusion of the internalized vesicles with the membrane to release the content into the cytoplasm. And we have found that there is a dependence of pH change that happens during this early endosome maturation process on the uptake and functionality of the received microRNA. And we also find the dependence of the mitochondrial activity of the recipient cells with the exosomal microRNA uptake and incorporation to the machinery in the recipient cell. So that much we have. So we have one manuscript on that particular topic, very exciting one, in review at the moment, and we hope that will be a very game-changing work in the field to say us something about the mechanistic detail of how the process may happening in the recipient cells. and what is the fate of the recipient cell microRNA. So with that, I just going to end by saying that they are the people that who work on this particular topic. I didn't had chance to touch the other aspects of the work going on in the lab. So the best possible way, the modulation of microRNA activity in the mammalian cells is the topic that we are working on. The regulation of intra and intercellular microRNA transport that are primarily I talked about that particular topic today, which is worked by Sudarshana, Chovik, and later by Komolika and Bottika. Komolika were on the HUR protein, Bottika worked on the internalization of the exosome in the recipient cell. Chovik did the polysome arrest work. Sudarshana was instrumental in working on the lipid-induced export of microRNA as well as cytokine IFN with the hepatic cell microRNA export and transfer occur. So later it's joined by a few others, talented students like Diptankar, Tushanto, Shoikot, and all others. And this path also, we play a very important role along with Onu to explore the PP2A pathway and some role of leishmania and cleavage of HUR and other process. So with that, I didn't have chance to very exciting work done by June to identify how the GP63 protein is also important to direct the dicer cleavage. And one of the first paper from my lab published in Cellulose Microbes in 2013. And I didn't have chance to also talk about the work of Shomi and Dipayan, who work on the neuronal pathway that affected by the microRNA, and the microRNA regulation happens in the neuronal cells, the amyloid beta exposed neurons and glial cells also. And also I didn't have chance to talk about the work by Jogaditya Bonishika or Moina, who work on very much importance of cellular organelles in controlling the microRNA biogenesis activity, a very different research niche that we have created based on the work they have done. But maybe in another platform, in another day, I was going to talk about that particular topic in more details. So we have some ongoing collaboration going on with Professor Edward Bartow's lab in Montpellier and also the Mihaela Zabolan's lab in Weizen from Basel, where we have two interesting projects that we are working on to have some interesting data in futures. So these are my primary funding bodies, from Human Frontier Science Program to the Wellcome Trust to the SARP India, Department of Science, CEFIPRA, and other kind of project funding organization, including the Intermodal Fund we have from the CSIR. These are the members who did a very good job. Some of them already left, and some are almost going to wrap up their work. Some are just joined, like Ishani. We have time maybe to answer some of them. So thank you again for inviting me to this exciting platform. And yeah, so I think you would have enjoyed to a certain extent the talk. Thank you.
Thank you, sir. We really enjoyed the talk. Thank you for the wonderful talk. We have more than 200 people watching us today, and we have a lot of interesting questions. So should we proceed to the questions, sir?
Sure.
So the first question from Abhishek Bhartan is, what determines which microRNAs are to be secreted through the exosomes and which ones will remain inside the cell?
Yeah, so this is not entirely known. So one can think about the different aspects that the export may affect. For example, I highlighted two things already. One is about the HUR binding to that particular microRNA that may affect the export of that particular microRNA to occurs. So this is one way of regulating it by specific microRNA binding proteins. By the interaction, they can affect the specificity of the microRNA to get exported. The other aspect could be the modification of the microRNA itself. So there are some modifications that causing some kind of post-translation modification, post-transcription modification you can think about, like methylation, UV generations and other kind of thing that make them substrates for the export to occur. So there is another kind of work that people have think about. The other aspect would be the localization of that particular microRNA in a specific compartment that is essential for the export to occur. For example, it need to be on the MBB membrane before the export should occur. So if there is some mechanism that can retain the microRNA in other compartments, that can also affect the microRNA export process. For example, if the substrate is there in higher amount, so that will increase the turnover of that particular microRNA, and that way the export of microRNA will be accelerated by the usage of that particular microRNA. So I think there are now quite a few number of proteins that have been discovered apart from HUR by other group also. La is another protein that people have identified as an important factor for the export to a certain class of microRNA. So that's why the specificity of the microRNA export probably primarily achieved through the interaction of the specific protein.
Thank you, sir. The next question is from Vyashak Vishwanath. How is the delivery of miRNA containing exosome to a different cell designed? Is it a planned action or an accidental event?
So that is a very relevant question in the field, and not much to answer at the moment. So people believe that there could be some specific receptor present on certain exosomes that make them susceptible to be taken by only certain kind of cells that have the receptor for that particular protein present on the exosomal membrane. But so far, we cannot really conclude on that particular topic or matter. So it can be speculated that there may be some specific protein factor present on certain status of exosomes that make them specific for that particular cell. But right now, we cannot say it's a very much orchestrated process. Rather, probably it seems to be a little bit random. But if it is orchestrated, it will be much stronger as a regulator. So I believe that maybe it will be explored in short duration to know that the exact way that may be regulated.
The next question is from Vijay Nagaraj. Are there any specific receptors involved in the uptake of exosomes in different tissues?
Yeah, more or less the same answer to my previous question, to the previous question as well. The receptor that may be present for the specific sets of exosomes received by the other cells.
Right. So the next question is from Abhishek Guho. Is it monoubiquitination or polyubiquitination of HUR that leads to dissociation of the miR122? But if it's polyubiquitination, then what is the fate of the HUR?
Yeah, so the ubiquitination pattern of HUR suggests it's probably mono and diubiquitinations rather than the polyubiquitination that we used to know for that take the protein to the degradation pathway. It is known that this kind of mono and diubiquitination is playing an important role for the cargo selections for the export to occur. So possibly this mono and diubiquitinated HUR not only ensure the unbinding of the microRNA, but also it's trafficking to the endosomal membrane for the packaging of the microRNA into the exosomes. So it's primarily the mono and di that we all observe is sufficient for the release of the microRNA from the HUR.
So can we, the next question is from Shanchari Gorai. Can we use LPS treatment in the case of chronic inflammation and for arthritis?
Yeah, LPS is there, I think, to increase the inflammatory process. So in case of anything, I'm not sure whether it will help to a certain extent to the inflammatory pathway. But it may be another way we can think about. If we have an anti-inflammatory molecule like GP63, which can target the HUR to degrade the HUR in specific context to reduce the inflammatory response. So maybe it is a good choice to target those HUR-like protein that are pro-inflammatory in nature or help the pro-inflammatory response to occur in the specific macrophage. So in that way, they are targeting by this GP63 is a part of liposome or in other means would be a very beneficial for this kind of chronic inflammatory pathways, including maybe cancer. Some inflammatory response is also there.
The next question is from YouTube. Sorry for all the technical interruptions that happened in YouTube today. You'll find the recorded version later on. The question from YouTube is there any report where microRNA can cause suppression of immune system in response to virus infection?
Yeah, the microRNA is known to suppress the immune response for certain mRNAs that are their targets. So one can think about to have some extra microRNA present there, or their expression get boosted in that particular cell type to reduce the cytokine expressions or cytokine storm that we are talking about. So that is an interesting thing to look at. And in that context, it's also important to know the fate of those proteins that are pro-inflammatory in nature, like H1 and others, in the viral infection context. And that may have a major role to play in controlling this kind of cytokine storm we thought about or know about in the viral infection context.
Our next question is from Deepak Kumar Podia. HUR knockout in mice is embryonic lethal. Have any of these HUR functions that you've described today crucial for the development or lack of it causing lethality?
Yeah, Deepak is one of the pioneers to show that there is a microRNA binding of HUR from Partho Sarathi's lab, Rai's lab. So I think, Deepak, your question is very relevant. But I think it could be the export of microRNA by HUR is a very universal process, as it seems, because it not only happens in the liver cells or in macrophage, but probably happens in the other cell also. So it's quite possible during the developmental phases when there's some kind of switching of microRNA need to occur. Some microRNA need to get turned off rapidly during the phase transitions of developmental phase transition, not the phase transition we're nowadays talking about. So, so in that context, possibly their role is there, and if they are not there, this kind of developmental phase transition may not occur, and that's why the arrest is there, and that's why it can affect the developmental process very much.
So another question from Abhishek Bhartan. Does the microRNA expression profile change during metastasis and localization of cancer cells as the cancer cells move from one tissue microenvironment and invade a different tissue microenvironment? And if yes, then how is this dynamic process regulated?
Yeah, I think it's known that they have a real change in the microRNA profile from one state to the other, as you mentioned. So this evidence is there, but the exact way they get regulated or they get misregulated is not explored. It's quite possible there are some kind of transcriptional contribution is there. There are some microRNA get transcribed in specifically one particular environment, not in the other. But there must be a way to also regulate the excess microRNA present in one context to the other context, because microRNA are usually stable. So when you're talking about the microRNA need to get lowered, you can think about the specific pathway that lead to the microRNA degradations, but it also there must be a specific pathway to reduce the microRNA in a rapid manner. In that sense, the export of microRNA is one of the choice that cells do have to reduce the burden of the microRNA, and that way it affect the export process.
So the next question is from Priyobrata Sinha. Is 4ABP1 the only mTOR agent to help free miRNA 122 from the polysome arrest? Is there any role of other mTORs like S6K1?
No, I think the other mTOR responsive protein like EIF40, BP, and S6K, we also have to find the S6K is getting upregulated with the mTOR activation in the context. But as the EIF4E directly have a role to play with the EIF4E, and EIF4E is known to play a role in translational boosting. And therefore, we say that probably EIF4E is a direct player in this context. But we cannot rule out that S6K also may have a role to play in the release of microRNA from the polysome, because it also can boost the translation process. You are very much right.
Our next question is from Renuka Dash. Is there any function of HUR relating to miRNA function in plant drought tolerance, especially in the case of maize?
So HUR don't express in plant, but probably there are other AU-rich element binding protein, because AU-rich sequence is also there in the plant mRNA. So there may be some other related protein may get expressed there and doing the similar kind of job. So we are not sure about that, but that is a good possibility that may need exploration to think about the microRNA transport that also occurs in plant between the tissues and the export of microRNA that may also occurs in plants. So there may be some similar kind of protein present there, but not known.
So another question from Abhishek Guho. AGO2 is a dsRNA binding protein. So how is it loaded to single-stranded miRNA of exosome in the recipient cell?
That's a very interesting question, but it is known that the AGO can bind a single-stranded microRNA as well. So this is, we explored and we find a dicer which is usually there to help the double-stranded microRNA loading to the Argonaut, help the Argonaut loading with the double-stranded RNA. So that is the process of the exosomal microRNA loading to Argonaut is independent of dicer. So that means you don't need the dicer to do the job. So retro pathway that actually affect the single-stranded loading of microRNA to the Argonaut occurs. So that's probably a way to also check the microRNA content or microRNP level in the cells, because some of the microRNA that are exported out as a single-stranded version may be retaken by the same cell and get reloaded to the Argonaut protein. So that's a way of buffering the microRNA level or microRNP level per se in the cells by this pathway. So it could be simple diffusions that ensure this kind of binding, but it could be some kind of other proteins along with the Argonaut that help the single-stranded microRNA loading into the Argonaut in the recipient cells, but not the cells.
So we have a few more questions. Should we continue?
Sure.
Okay, sir. Sir, Shondipan Josh is asking, amino acid starvation deactivates the mTOR complex. So conventionally, it can arrest protein translation. How does it work here?
No, the mTOR pathway didn't express in the hepatocytes context or the starvation context. Rather, we see the growth arrest context, we actually follow the mTOR pathway. But mTOR get reactivated during the refed context. So possibly at that context, the export need to occur more strongly because it need to revise the microRNA pool there also. So in that context, maybe the mTOR pathway may boost the export process. But that's not export. I should not comment on that. The only thing we can say that the mTOR boosting in the growth arrested context when the microRNA level is high actually help the microRNA export to occur via the activation of the mTOR pathway.
So the next question is from Shondeep Pramanik. Why does the GP63 target HUR? Is there any basic reason behind this?
The molecular basic reason is not known. So because the HUR specificity, GP63 specificity for its substrate is little bit ambiguous, because some protein are targeted very preferentially, some don't. Like Argonaut doesn't get affected, but our dicer get affected. So I think the localization of that protein and the its proximity with the GP63 that are also maybe a part of the vesicles called phagolysosomes, where the parasite resides, is probably a factor that can control the protein and protease interaction, the GP63 and the protein interactions. So that's where the specificity comes from, the localization of the protein in the proximity of the vesicles where the leishmania resides. That can also make a difference, but I am not sure how the specificity may ensure, because in vitro also we have the specificity that cannot be there with the vesicles thing as such. But the molecular details of that thing is not known, because the protein is a little bit difficult to get cloned and expressed. Such that the its specificity study has not been done in very detail.
So the next question is, can miRNA under any circumstances cause upregulation of gene expression of pro-inflammatory cytokines and have some contribution in autoimmune disorders?
There are some reports that suggest that some of the microRNA that present on the cytokine sites can act as a positive regulator of translation, which is a little bit, I would say, controversial in the field, because there are only couple of labs that published that particular aspect, and no much follow-up was there after the publications was there. So, but it could possible that this microRNA binding maybe have a regulation on the mRNA stability, because maybe by binding the microRNA, they get stabilized to certain extent, because their stability is not only regulated by the H1 in a positive manner, but there are protein like AUF like proteins, they are there. TTP and AUF, they are the negative regulator of the stability of the mRNA, which has the AUF sequence. So once they bind, they probably induce the degradation. But it may be possible that the binding of microRNA and the Argonaute proteins may ensure the stability of the mRNA against those RNA degrading enzymes and proteins like AVF1 and TTPs that are also countered by H1. So that's why maybe they have a positive role to play by storing some of the microRNA targeted messages in a specific compartment where this protein cannot have an access. And that way, apparently, they help the upregulation of the gene expression from those things.
So Dr. Sanjeev Pandey is asking, are the microRNA containing exosomes, do they have anything to do with the origin of enveloped RNA viruses?
It's not probably the similar thing, because usually the microRNA that are released are very small in size, whereas the viral RNA are usually very big in size. In principle, they have some similarity in between, because the way they traffic, the way they're taken up into the cells. So some of the pathway may have some commonalities in between. So possibly this exosomal pathway is more ancient, and then the virus evolved to adopt that pathway to get into the system and use that particular trafficking pathways to enter into the target cells and utilize the machineries present there. So in that way, they are probably adapting the similar kind of machinery that the exosomal pathway also adopt. So that may have some similarity in that way.
So we'll have one more question before we wrap up. MicroRNA is considered the new target for small molecule drugs. So do you think this is a good venture for medical research, and what are your views on it?
Yes, I definitely think so. Not because I am a microRNA biologist, but as this is a very much important regulators in different contexts, and you can see the difference of expression of the microRNA is there in different disease contexts, including cancer and other kind of diseases. So it is very much possible that these microRNA are trafficked from one cell to the other and make a balanced expression of genes in different cells. So one can think about targeting the specific microRNA at different steps, not only at the activity level, like inactive the microRNA, but also can prevent the trafficking of microRNA between cells. So as it is known that the trafficking of specific microRNA from one cell to the other is playing an important role in certain disease development, particularly in cancer context. So if we have a small molecular inhibitor of those microRNA or the trafficking process itself, then that could be a good way to target those diseases that are otherwise difficult to target.
So thank you for answering the questions. Thank you for a wonderful talk. It was a pleasure listening to you. Thank you everyone for watching, asking questions. To all those whose questions could not be addressed, apologies. I would like to now turn it over to Professor N.R.A. Banerjee for the vote of thanks.
Thank you, Suvendra. That was such an exciting talk and such stimulating round of questions. So thank you for addressing them in such great depth. And you know, at the end of the talk, I was looking at that picture of your lab members, and I was reminded of the other slides previously where you were talking about the biogenesis and the sequestration and targeting of the microRNAs. And I was marveling at how knowledge is added little by little by the members of the science faculties.
Yeah.
So I hope the attendees of this session are going home with food for thought from Dr. Bhattacharya's wonderful talk. And thank you, Suvendra, once again. And I do hope that you will come back in the near future and tell us more about the unfolding stories in your lab. So thank you very much.
Thank
Thanks
you.
to the team. There are, unbeknownst to the people attending here, there are 7 people working in the background, including Rupkatha, who is visible to you. The others are not, and they are sitting in different time zones and working throughout the talks to bring it to you uninterrupted. However, we apologize for the little technical difficulties that we had during the session. But Shubhendra's talk, I think, more than made up for the difficulties. So thank you once again, Shubhendra, for sharing your exciting science with us. And thank you everyone who attended today.
Thank you. Thank you. Bye.
Bye.
I really enjoyed.
Thank you, sir, on behalf of the PPV team. Thank you, ma'am. So today, I hope you enjoyed the session today.
Looking forward to it and have a good day. Thank you.